Business meeting. It's my pleasure to be hosting the last session of the meeting. And first up is Alexis Huet.
Perfect. Well pronounced. So I will speak today about an old friend, which is HK97. In particular, I will speak about the early stage of assembly, and in particular, the role of the portal in the regulation of the earlier step of the assembly of the Capsid. So basically, what is HK97? For the one that doesn't know what is HK97, it is a SIFO. It has an icosahedral capsid, and all the capsomer penton and exon are composed of the major capsid protein, composed by the eponymous HK97 fold. But the major capsid protein is not only the HK97 fold. The major capsid protein also has about 100 amino acids on the N-terminus, a scaffolding domain that is bound, which means that actually during the assembly, the stoichiometry of the scaffold compared to the major capsid protein is 1:1. Also, of course, the capsid is not fully icosahedral. So the symmetry of the capsid is broken where the portal will bind, where the tail will attach. And generating this problem here that has been called for a long time, the symmetry mismatch, where the 12-fold portal bound somewhere in a 5-fold environment. The pathway of HK97 has been well defined, and basically is a major capsid protein together with a scaffold, plus a protease, plus a portal that goes together from a ProEd 1. Then the scaffold gets removed from a ProEd 2, and the terminase put the DNA in the capsid, and after expansion from the final capsid, which is prepared for the virion. What is very interesting with HK97 is that it's a very modular system, which means that if you express only the major capsid protein with a portal, you get a ProEd 1 with actually all the scaffold domain, since actually is attached with the major capsid protein. And interestingly, when we produce this ProEd 1, when we purified it, what is observed is basically that actually the ProEd are making oligomer of ProEd. So the ProEd can be either dimer, trimeric, and all this polymer of ProEd are attached together by the portal and are detergent sensitive. And if you look at the same sample, the by cryo, actually, you will observe the same kind of oligomer. You can see here you have this kind of thing that is attaching finally all the ProEd together through the portal that you cannot see here, and here you have also oligomer. So since we have the cryo, we obviously did the reconstruction of this object. So both at the regular vertex as well as at the portal vertex. So at the regular vertex, we see the exon, we see the penton, and we see under the exon and under the penton, there is this blurry density here that we assign and we consider thinking that this is a scaffold. So, and at the portal vertex, of course we see the portal, but we see two other things here. We see a blurry density on the top of it, which we believe is a detergent-sensitive product. And on the side of the portal, there is two structures here that are composed of alpha helix that are going along the portal toward the center of the capsid. And we believe this is a scaffold. When we look more carefully to the density here, actually what we observe is indeed we can assign every single part here. So in yellow will be the part of the major capsid protein that actually has the HK97 fold. In green is a scaffolding domain. And in red is a portal indeed. So, and what we see in grid here is that actually the scaffold is present in two positions. One which is very close to the capsomer, and which is very rigid, but which can go at very high resolution and is completely bound. And another one that actually, this extension that is going toward the center of the capsid, that actually is making a coil of 3 helices. And out of this coil, actually there is 10 of these coil of 3 helices. So if you want to look how it works here more carefully and more in detail, is that actually every single coil here originate at one capsomer. So there is 5 capsomers around the portal. And then from every single capsomer, there is 2 coil of 3 helices each, which means that actually every single helix of the coil originates from one subunit of the major capsid protein. Also, this explains why, actually, these call actually are spread around the structure asymmetrically. Because in that case, here in that direction here, you have 6 of these call. And in that direction, you have 4 of this tower. And because, actually, they have to follow the pattern here of the capsomer. And at the end, because here is the question, here we have a structure, and the final question is, why is the symmetry mismatch? And then when we look at this structure that finally is made of call, is actually what is seen is that the call, which is a scaffold, which is also the major capsid protein, finally adopts a pseudo-12-fold conformation. What is happening that you have this asymmetrical distribution here of 6 towers, here of 4 towers, and here you have 2 spaces that are completely empty, which means at the end, the call actually interact with the portal at every single time at equivalent position with a pseudo-12-fold conformation. Also, and which is also important, the scaffold here that again originates from the capsid protein, which is a major capsid protein, actually is the only one part of the major capsid protein in the ProEd 1 stage that does interact with the ProEd. And it's like the scaffold itself is making a buffer between the portal on one hand and the shell on the other hand. And which is confirmed here once if you go to the section and you look at the density along the section, actually we can see that all the density that we assign to the scaffold actually systematically interact with the red density of the portal without having any part of the yellow density that never interact with the portal. So, at the end, then, in HK97 fold, the fold of the HK97 is is a bit more than the fold that we know. Is the major capsid protein itself finally is a combination of both the fold plus the scaffold that now we can have some good idea about its conformation. The scaffold originates where actually just after the end arm, the cleavage site actually, the end arm is in the cleavage site is completely diffuse and accessible, probably leaving the position for the protease to be to cleave. And after that, the scaffold starts just after the end arm, where actually the end arm is in the ProEd 2, and then goes along with the strand, turn, helix, then the spacer, then the helix that will form the coil. So, now that we have some idea about what the portal does in terms of dealing with the symmetry mismatch, having the pseudo-12-fold structure, and finally being between the portal and the shell, the question is, what happens when there's no more scaffold then? So, and when there's no more scaffold, what's happening is that probably also this part here that is blurry disappear, and then after that, the terminals can start to work and start to package. Then, using the same easy system, we can easily now adding the protease in the system, producing the ProEd 2, and doing exactly the same reconstruction here with the 12-fold input symmetry, simply opening the ProEd, the portal of the ProEd 2, with all the well-known and characteristic system. What is interesting now, if you look here at the 2 portal reconstructed with the 12-fold input symmetry here of the ProEd 1 here and the ProEd 2 here, is that beside the scaffolding domain which is there, actually the density are almost strictly the same, except for some fluctuation in terms of density, which means that some part might be more or less flexible. But in terms structurally, I cannot see any differences, which is confirmed here when I superimpose the 2 structures with the 12-fold symmetry. We are in red of the ProEd 1 and in blue of the ProEd 2. But, and also, yes, also at the level of the clip domain, I cannot see any differences here. The ProEd 1 and the ProEd 2 doesn't see obvious differences. So the clip domain doesn't follow a 12-fold distribution without symmetry imposed, and it seems that the ProEd 1 and the ProEd 2, removing the scaffolding domain seems to change dramatically the conformation of the clip domain. But when we do not impose symmetry, what we are seeing here is that actually the shell here, that is one exon, that is another exon, actually, which is there close to the portal, is getting closer here to the wing domain of the portal in the Parade 2. Here, we have the same kind of idea. You have this part here that is going up and going close to the wing domain. And on the top, having this stuff, there's something here that is interesting that is appearing, that this density here that is appearing. That actually is not there, more or less where the scaffold was. And then it appears that this density, finally, is this yellow density down there in the Parade 2, and actually that we can see that is here, that seems to be exactly interfacing itself with the shell. So again, having another structure that is flexible in the Parade 1, that is getting rigid in the Parade 2, that does interface between the portal and the shell. And it appears that this part is actually the end terminus of the portal itself that is starting to, that is getting organized in the Parade 2, and then seems to somehow play the role of the scaffold in the Parade 2 in order to compensate for the mismatch. So, to conclude, actually, the fact is that between the Parade 1 and the Parade 2, and why this detergent-sensitive thing is removed and the terminals start to bind, is not obviously because of the clip domain and change of conformation induced by the removal of the scaffold. It seems more likely that it might be from actually some slight reorganization of the capsomer around the portal and a global change of environment of the portal after the removal of the scaffold. Thanks.
All right, any questions?
Oops.
So would you say then that the interaction between portal and the major head protein is weaker in the ProHead 2 after you cut out the scaffold?
Absolutely. But actually, you can see here actually without reconstruction. Actually, that is something I didn't say. But here's the shell without symmetry imposed. The shell is extremely solid here. And here, actually, we are losing the resolution extremely fast, which because actually the portal in the Parade 2 has a lot more freedom than in the Parade 1, which, again, the Parade 2, the thing is that it's a transitory step that is probably not even existing in vivo, because Parade 2, as soon as the scaffold is removed, I believe the terminus bond. So in that case, it's a different species. So Parade 2 is it's kind of a mistake of assembly.
So the portal can spin then.
One. Yeah.
Any other questions?
So what's going to be the effect of having some DNA in this structure?
But to start with, no, inside, you mean inside the portal or inside the capsid? Or both?
The capsid in ProHead 2.
Right now, I don't know. First, it will expand. I wish to know what happened. First, you have to think that you have the terminals, then you have the DNA coming, and then it expands. And when it expands, it's still packing. So having the expansion is not stopping the terminals to bond. So what stops the terminals to bond when the DNA is in it is an open question.
Okay, great. Thanks so much, Alexis.
Yeah, you're welcome.
All right. Oh, here's the. And next up, we have Charles Bridges, and he is going to talk to us about the P22 capsid maturation.
Yeah, so thanks for having me. I appreciate being able to have the opportunity to talk. I'm actually going to talk more about the genetic regulation of capsid assembly today, so a little bit of a switch, not a total falsehood, and I'll explain why. So I work on salmonella phage P22, so I don't have to describe to this audience much about it, but it's a T equals 7 phage, double-stranded DNA of about 42 kilobases, and it uses the HK97 fold. So the general P22 assembly pathway begins with nucleation, so recruitment of portal protein by scaffolding protein that forms a nucleation site here, which then scaffolding protein can recruit coat protein to oligomerize around that nucleation site. Some ejection proteins are incorporated in, and then terminase complex is able to bind, package the double-stranded DNA, upon which scaffolding protein exits the capsid. And tail machinery is added for a mature virion. So I work on a few things in the Teschke lab, one of which is kind of in vitro assembly of the phage genome, so we can kind of high throughput mutations. I'm also working on in vitro DNA packaging, so some of this work we have down pretty well. The actual maturation process is much less well understood. And I'm also working on dissecting the P22 genome, at least the structural gene cluster a bit, to make synthetic PLPs and virus-like particles. And it's this work where I dissect the structural genes into functional modules where some of this other work is kind of popped out. And so, for example, module 1 here is the terminase complex and other genes that are upstream of those. Module 2 is like the typical procapsid complex, so coat protein, scaffolding protein, and portal protein. And then I've got a plug complex, an ejection complex, and tail is on its own separate operon in the P22 genome. And so by doing this, the hope is to eventually be able to fully control what kind of output particles we get. And so, for example, if we wanted to package competent procapsids, we would use modules 2 and 4, where there's a procapsid assembler, and then also all the ejection proteins are present. And eventually, when we get the in vitro packaging to work a little bit better than we have now, we can actually start to package some of these PLPs. And so by dissecting up the genome, we're able to fully control which proteins are present during assembly. And also, we gain some insight into the genetic regulation by teasing those interactions apart by dissociating them and genetically unlinking them. So the very first thing I tried with this was expressing module 2. And so in this instance, we've just got portal protein, scaffolding, and coat protein under the control of the T7 system. And so the first thing I noticed in this type of setup was that portal protein is overexpressed. And so when I express modules 2, 3, and 4, which are the procapsid assembler in the presence of the plug proteins and ejection proteins, I can see that portal protein is still overexpressed. Over down here, we have coat protein and scaffolding protein. And coat protein seems to be somewhat downregulated in the presence of the plug and ejection proteins. And so what this suggests is that perhaps coat expression is modified or regulated to some degree by having the ejection and plug proteins downstream. And so one of the things that we'll do later on when we have the in vitro packaging working is to co-express the terminase system with these other plug ejection and procapsid modules. And so just to try it out, I expressed all these modules together and immediately noticed that portal protein was way down-regulated, as well as coat protein and a pretty significant decrease in scaf expression as well. And so what this suggested was that there's some genetic regulation of the procapsid components that are present in the upstream terminase sequences or proteins. And so the stoichiometric ratios of these components are very important for procapsid fidelity. And so what I did here was take the PLPs, so these are in vitro expressed, put them over a sucrose gradient and analyze the peak fractions by SDS page and TEM. And what immediately popped out is that it's not just the expression patterns that overexpress portal. These particles actually contain more portal than would be expected by wild-type procapsids. And another thing is that scaffolding proteins decreased 2 to 3 times per PLP. And the PLPs also contain more aberrant particles than would be expected from a wild-type procapsid prep. So we've got more spirals and rosettes than we would normally see, and also more T=4 petites, suggesting that this is a scaffolding regulation mediated phenotype. And so these aberrant particles we attribute to altered stoichiometry. And this has been observed before by the privileged group, where they overexpressed portal protein during an infection and saw proheads with 2 portals incorporated. Here they were observing this because there were tails present that could attach to those 2 portals. Sherwood Cashins and Jonathan King and others have previously demonstrated that scaffolding protein is able to auto-regulate its expression post-transcriptionally. So here on this mRNA, you can see that too much scaf will actually cause a binding to the mRNA and downregulation of scaffolding protein. And so I was curious if there are other intrinsic regulation, other signals of intrinsic regulation of portal, scaf, and coat proteins beyond just the scaffolding regulation that we know about. And so I was looking for things like repeat regions, internal promoters, transcriptional attenuators, and RNA secondary structure. And so I kind of went diving into the literature, and one of the first things I came across was a transcriptome by the Pinedo group here in the UK. And they were looking at lysogeny and transduction, and so I kind of just repurposed their RNA-seq data for my own use. And so I mapped their transcriptome data. This is 30 minutes into infection. So I mapped that to our structural gene cluster. And there's a couple things that kind of popped out. First, the transcription levels start relatively high and kind of peter out over time. And that's a good thing. That's consistent with RNA polymerase dissociation over a long distance. But then I also noticed some things. So this RNA is negative regulated. These are some regulatory genes, as well as the holins, which are not expressed early in infection because you don't need those until the cell's ready to lyse. And then also, quite strikingly, scaffolding protein and coat protein were about 5-fold upregulated compared to portal protein. Now, this is not consistent with a single long transcript. And so here's a zoom in. So I did some bioinformatic analysis, and I found some putative promoters that sit upstream of scaffolding protein in the 3' end of the portal gene. I also found a stem loop at the end of gene 5, which is conserved between HK97 lambda and P22. And so this kind of got me digging a little deeper to see if there were other regions that could contain RNA structure, secondary structure. So this is kind of a busy slide, but what I'd like you to take away from it is that this is the portal gene here. And so the region that I was most interested in is right up here. This is the ribosome binding site region. But if we go backwards into gene 2, or large terminase, we see all these, every one of these black gray boxes is a sort of TGA-ATG motif. I don't know the significance of that, but it got me digging a little deeper for patterns. And I found all sorts of direct repeats, as well as a stem loop here in purple, and some larger regions of putative RNA structure in red. And so what I did was focus on the immediate upstream sequence from portal protein. And so here's the portal ribosome binding site. Here's the portal start site. And here's the stem loop that I was talking about, shown here. This is just an RNA fold representation. And use some other bioinformatic tools to find potential transcriptional attenuators. And there was a pretty strong hit right upstream from the stem loop. And so the first thing I did was make some plasmids to test this hypothesis that these RNA secondary structures were regulating portal protein synthesis. And so here you'll see this key over and over again. So this is just the T7 promoter. Now I've switched to using the portal ribosome binding site and expressing GP1, GP8, GP5 portal scaf coat. So Here's a gel showing those results. The first one is the original procapsid assembler using the T7 ribosome binding site. And portal protein is downregulated using its own native ribosome binding site, which would also be suggested based on the sequence that it's a weaker ribosome binding site, and that is shown to be true. The upstream, or the putative upstream RNA secondary structure in GP2 did not appear to further regulate GP1 expression. And in the presence of GP1 ribosome binding site and coat protein, scaffolding protein is increased about 2 to 3 times. And this corroborates observations by Castjans and King in a couple of papers that showed similar thing. And so now I removed coat protein from these constructs, so same exact plasmids, just no coat protein expressed. And what we can see is that portal protein is still downregulated. There's still no significant alteration in expression using that secondary structure or any of those differences between those plasmids. And so our general hypothesis that the RNA secondary structure regulates portal expression, at least regarding intrinsic regulation, is probably not correct. And so this kind of moved me to genetically unlink these two plasmids and see if we could determine if there were differences between portal protein expression binding, scaffolding protein expression binding portal RNA. And so in this case, I co-transformed portal-only and scaffolding protein-only plasmids into the same cells, took a bunch of clones and expressed. And what we can see is this is clone 1 through 6. So this guy only expresses scaf, this both, and something like this would only contain portal protein single plasmid. And so when portal and scaffolding protein are co-expressed, both proteins seem to be downregulated, suggesting that scaffolding protein doesn't just bind its own RNA, it may also regulate portal protein expression. So another thing that I noticed was that there were differences depending on if coat protein was present. And so in the absence of coat protein, and thus no chance of procapsid assembly, portal protein expression is increased, suggesting that there's some relief of negative regulation, perhaps by coat protein or perhaps by regulation of scaffolding protein. And scaffolding protein expression is decreased 2 to 3 times, suggesting self-regulation. And this, again, has been seen by Castjans et al. In the mid-'80s. Now, alternatively, in the presence of coat protein and thus procapsid production, we see that scaffolding protein expression is upregulated, probably due to the sequestration of scaffolding protein into those procapsids, and thus the need to increase expression for more scaf. Portal protein is downregulated, probably to prevent that aberrant particle formation during in vivo assembly. So we thought we'd go towards more direct evidence for scaffolding protein interaction with RNAs. And so the method we used was in vitro transcription of portal, scaffolding, or coat RNA. And so these are single coding frames on a plasmid in vitro transcribed with a T7 system. And then we UV cross-link those mRNAs with scaffolding protein. So in this figure, mRNA will be in green and scaffolding protein will be in cyan. So first, to control scaffolding protein alone, every set that I show you will have an uncross-linked version and a UV cross-linked lane. And so the first here is RNA alone, so un or cross-linked, and then scaf plus RNA, uncross-linked or cross-linked. And so in this case, scaffolding protein was cross-linked with GP1 only mRNA, and you can see a shift upward in the RNA that co-localizes with scaffolding protein. In the presence of GP8 RNA, scaffolding protein, most of it hangs up here. You'll have to take my word for it. There's a little bit down there. We see a smaller shift, and we see a smaller amount of co-localization. And then if we try to cross-link scaffolding protein to coat RNA, we see a smearing, but we don't see a huge shift, and most of the protein colocalizes up here. And we see some of this type of thing with our negative controls using albumin as well. And so, in summary, capsid assembly is regulated at multiple levels. Stoichiometric ratios of these scaffolding, coat, and portal proteins are imperative for procapsid fidelity. The single late transcript does provide seemingly a baseline level of transcript for all the structural genes, but internal promoters may increase the scaffolding coat ratio, and also allows more nuanced regulation of scaffolding protein levels in the cell. And scaffolding protein may additionally regulate portal protein expression by binding portal RNA. And taken together, scaffolding protein may be acting as a sensor and regulator for procapsid fidelity. And so the last thing I'll show is a very rough model. And so on the bottom here is P22 genomic DNA. So this is the P-late promoter from which about a 20 kb transcript is produced. Some regulatory stuff up here. This is small and large terminase, portal, scaf, coat, plugs, and ejection proteins. Tail protein is down here. Tail spike's on its own operon. And so these are the putative scaf promoters that I found here. I didn't talk about it today, but there's some hand-waving preliminary evidence that scaf may interact with this DNA site as well, and here's the terminator. So here's our P-late transcript producing all of these proteins. And what I'd like to point out here is that portal protein showed the strongest binding scaffolding protein showed the strongest binding with portal RNA. And so this seems to be a tighter association, maybe with lower dynamics. And then additional scaf transcripts showed that scaf actually interacted with its own RNA in a weaker association. And this sort of makes sense if you consider that the free scaffolding pool is being recycled through procapsid and phage states. And so you'd want to have a weaker association here so that it could have a more nuanced regulation. So we call this a loose binding and high dynamics. And I didn't get to talk much about it today, but there's some preliminary evidence that ejection and plug proteins may also be regulating coat protein expression. And there's also even a little bit of evidence, and this is all hand-waving speculatory stuff, that coprotein could be interacting with portal RNA as well. And with that, I'd like to thank the Teschke Lab, my advisor Carol Teschke, the rest of my group, and again, the PVA organizing committee. And I'll take any questions.
Beautiful stuff, Charles. Just in a general sense, how tight does this regulation have to be? I mean, you sort of have some insight into that based on the fact that you're looking at different expression levels.
Yeah.
Is everything fine as long as you stay within, 2 or 3-fold of the ratios that seem to work, or does it need to be like 10 or 20-fold before it starts being driven off pathway?
Yeah, that's a great question. We haven't done any of those types of studies. This is all very preliminary data in the last month or two. And so I would speculate that scaffolding protein expression has some wiggle room. What we've seen so far, portal protein expression has to be really tightly regulated or you get all these aberrant particles.
We have time for one more question. The rest of you can attack him at dinner time.
I have an historical.
Paolo is a gentleman. Is the concentration, the intracellular concentration of scaffolding protein, what do you think? Is it constant or does it go through different concentrations depending on its actual function during assembly?
Yeah, so there's only a couple papers on this that are, quite old. And from what I understand, the cell or the phage is trying to maintain scaffolding protein concentrations as best it can, and so I think the concentration is I won't even say, but I think it's trying to maintain as best it can through this genetic regulation, trying not to fluctuate. Any deviation for any amount of time would start to produce aberrant particles.
All right, thanks, Charles.
And I see we've saved the best for last. 14 minutes is going to go off.
14 minutes, it's going to go off. And when do you start getting threatening? You have to.
At a minute. Okay.
What?
It's my pleasure to introduce Bob Duda, and he's going to talk to us about Hong Kong 97 Capsid scaffolding domain.
Yes, well, welcome to you all. I'm Bob Duda. I've been working on HK97 for a number of years, and I'd like to tell you, as she said, about some of the experiments were done investigating the function of the scaffolding domain. So I don't know the rules here. So that's the laser. That must be to the right. Okay. So Alexis went over the pathway, and many other people have gone over the assembly pathway. So I will just say, we make a prohead, we make a procapsid. We get DNA package, DNA goes in, we get a mature capsid, and all sorts of interesting things happen along the pathway. But I'm going to concentrate on the first step. And this is a generic pathway where you could have a separate scaffold protein as opposed to the tethered scaffolding domain that HK97 has. So HK97's scaffold domain, as Alexis pointed out, is stoichiometric with the major capsid protein and packaged, so it's always packaged inside. And once assembly is complete, the protease is activated by some mysterious mechanism. You get expansion to Proa 2 and you make a head. So scaffolding proteins have a number of functions. So I've just listed them here and I'll just go over them really quickly. So they're properties anyway. So they're upstream of the capsid gene usually. They're present in the immature capsid and absent in the mature. They're sometimes predicted to be flexible or have intrinsic disorder. They're also predicted to be largely alpha helical, and in the case of HK97, no matter how we measure it, the scaffold protein has been biophysically been measured as being alpha helical years ago, and you've seen evidence that it is alpha helical actually in the particle. It plays roles in capsid or viral scaffolding proteins play a role in scaffold size and scaffold shape regulation. In the absence of a scaffolding protein, you get abnormal assembly. And then there are special conditions where the scaffolding protein might influence the capsid size, depending on which system you look at. You also have a role in portal incorporation. So as Alexis pointed out 2 seminars back there, in HK97, the portal binds to the scaffolding domain, and that's how it gets incorporated. And the scaffold proteins bind to the interior of the procapsid shell. That's how they assist in facilitating assembly, and in other systems they even have other roles, which is to help incorporate additional proteins which will be injected. So in P22, which people have been talking about, the genetic analysis, and this is from a rather old paper, has shown that the functions are distributed along the length of the protein. And so you have some functions mapping to the N-terminus and portal recruitment, and coat binding mapping to the C-terminus. And in many ways, the HK97 scaffold protein has some similar properties. So here's a summary of a whole bunch of mutations that were mostly made by one of the people I listed on the title slide, Bonnie. Bonnie O was a graduate student in Rogers' lab. And so we have mapped the C-terminal domain is the part that binds to the interior of the capsid, and sort of, so it's, and mutations, point mutations in that domain lead to abnormal assembly, which is sort of like loss of a scaffold protein in other phage systems. We have portal incorporation mutants mapping in the middle. We also have some data about protease incorporation, which I'm going to not talk about today. And Bonnie also made, yes, okay. Let me continue here. So the other thing about the scaffolding domain of HK97 is that it was long ago predicted to be largely alpha helical for the first like 70 amino acids. And so coiled alpha helices. So coiled coils are easy to recognize because they have these special properties. The 7 residues in the alpha helical repeat. The A and D residues are hydrophobic in coiled coils, and so it's easy to recognize how they're going to come together and whether they'll have a propensity for that. And so indeed, in it having been predicted to be alpha helical and coiled, as Alexis showed you, at least in the scaffolding domains that are binding to the portal, you see 3-stranded alpha helical coiled towers. So, and there's actually additional evidence for similar coiled towers, both in HK97 mutant and in a couple of larger phages that we work with. So in a peculiar mutant of HK97 that happens to make small procapsids and normal-sized procapsids, the pentons are visible as coiled towers underneath the pentons. And so we don't see this in the wild type situation. So, but that suggests that pentameric coiled towers might be a part of the assembly pathway, but that in the normal situation, they're too unstable to visualize. And we also see similar what we interpret as coiled towers, as Anna showed you earlier in her talk, under the pentons. In addition, we see what we interpret to be 6-stranded coils underneath the symmetric pentons in the middle of the face of the T=9 capsid. That may be a little too much detail for you, but if you ask me questions about it later. But anyway, so the argument is that the N-terminus of the HK90-SCAF7 capsid protein has this tendency to make alpha helical coiled coils probably of various kinds, and they may have a role in assembly. So Bonnie made deletions in the N-terminus, and most of them were absolutely uninformative and boring. That is, most of them just made absolutely nothing. There was one mutation of about, I think, 11 amino acids near the N-terminus, and it still assembled, although rather poorly. And it also incorporated portals, and most of the particles that we saw appeared to have portals. Deletions in the middle were all dead. She made this other mutation. She made 2 other mutations that, taking this prediction that we had at that time, she removed the part that was predicted to be not alpha helical. She called it delta first hinge and delta second hinge. And delta first hinge was a bust. Delta second hinge actually made particles, but that deletion is the one that made the mixture of T=4 and T=7 particles. And we actually don't understand why that's happening, and we would like to. And that mutant del2H removes 7 residues and still assembles. And that's as I will show you in a second, that mutation will be removing this sort of linker region that Alexis described between the scaffold binding domain and the long helical towers. The reason this is shown as density is because we don't have good enough density to model the side chains in there. So armed with Bonnie's result, we decided, what the hell, let's just make every size deletion you could possibly imagine starting from that point. And we were totally shocked at the results. So we made deletions all the way 1 through 13, and then a few more, and then one that was 26. And this is the kind of gels we analyzed this on. I'm not going to dwell on this too long. Alexis gave me an introduction that I didn't realize I was going to get. So he pointed out that the ProHead 1 with a portal, they're sticky. They're stuck to membrane components, and they make dimers and trimers. They do that so efficiently that is an assay. We can use that as a very convenient and quick assay. If we see this band on an agarose gel of dimers or even trimers, then we know that the portal was incorporated in that. So we can screen mutants. These are just samples of crude extracts from 1.5 mil cultures. And so we can look, and basically the take home is that we can remove one amino acid, and that's what's on the end here. The first one here removes one amino acid from, let's go back here, one amino acid from this section, right? It's probably shifting the tower down a little bit. And it still incorporates a portal, and it still assembles normally. We haven't tested this by complementation, just haven't gotten around to it. And then as you go up, I will just say that we can tell whether the small capsids are being produced because there is this is the ProHead 1 procapsid band, and if you see a faster moving band, it means the small T=4 particles are being made. So this is just a quick and easy screen for what's going on. And for some of these mutants, this is as far as we've gotten. And so this is sort of a summary that portal was incorporated if you deleted one amino acid, but if you deleted any more than one up to somewhere around 10, there was no portal incorporation. At larger deletions, you started to see a hint that portal was incorporated a little bit. And my guess is that what we're doing when we make these mutations is we're shifting the towers down by shortening the base. And so at some point, up in the higher regions of the tower, there may be pseudo portal binding sites that are being utilized in some of these other mutants. We haven't done any modeling to try to understand what the basis of that is yet, but I had been planning to do that for a long time. But anyway. The other thing is that there's a peak of making small particles at 7, and then after 7, it goes to making normal particles all the time, and then some of the higher ones, you get a few small particles again. And so this is trying to tell us something about size regulation, but I don't know what. So we have a good correlation between the mutation results and the phenotypes of the mutations and the structure that Alexis has shown you. So we have the interior capsid binding domain, and mutations there are affect assembly. We have mutations in the middle, which affect portal incorporation. And we have, as far as that goes, we can remove up to 26%, which is 26 residues, which is about 1/4 of it, and it still works. So that's suggesting maybe that the N-terminus is really important for assembly. So here's my sort of quick summary. So what have we learned about its role in particle assembly? Well, the N-terminal scaffold region is sufficient for particle assembly because we can delete from the middle up to about that level. The central portion is not required for particle assembly, which was a surprise to all of us. So that's suggesting that maybe the N-terminus is really important for initiating the formation of capsids, maybe by initiating the formation of capsimers. So this is my very crude model where this data that the N-terminus of the coiled coil is absolutely essential for assembly suggests that maybe the mechanism is that maybe the capsimer subunits are brought together initially by that kind of interaction, and then the body of the capsimers are brought together to align and form a capsimer. So that's a model that I keep in mind. As far as its role in portal incorporation, we know from Alexis' structure and from the mutation that this central region of the portal is absolutely essential for portal incorporation. But there may be alternative interactions between the portal and the scaffold that are possible if we shift that tower so that it's the residues that are near the N-terminus are now down near the portal. And what about particle size and shape? Well, so as I said, there's abnormal assembly if you mess with the interior capsid binding domain of the scaffold. There are these deletions that shorten the spacer and linker of and then the tower region can lead to smaller particle formation, and that is still a mysterious thing, but we hope that if we can get some good high resolution structures, we'll get a little more clues to make some mutants that are a little bit easier to interpret. And we also, that same removal of the spacer somehow seems to stabilize this alpha helical coiled towers both in the small particles and in the wild type version in that mutant. So what's next? Well, I would like to determine the extent of the requirement for the N-terminal part by continuing the series of deletions. We would like to make more mutations. We would like to learn a little bit about the scaffold and the portal incorporation to see if we can maybe determine structures of some of these things that seem to bind and reveal an alternative portal binding site. We're hoping that we hope that we can use these deletions that stabilize those penton towers to get some structures in which we can see side chains in the towers, which we haven't been able to do yet. And we also want to look at the role of how protease is incorporated, which I haven't talked about, and is an interesting by itself. So I'd like to thank you all for your attention. This work was started when Roger Hendricks was still around and I was in his lab. Bonnie did a lot of the mutagenesis, except for the recent work, which was done by Josh. And Alexi and James did all the reconstructions that I've shown you. And we've had many collaborators over the years, both from Alistair Stevens' group, from Jack Johnson's group, from James when he was in either in Alistair's group or when he was in Grenoble, and since then in this Department of Structural Biology. Okay, thank you. Good. How did I do?
Okay, we have time for a couple of questions.
Hi. In building up this 5 or 6 helix bundles, which are all parallel, you are increasing enormously the dipolar moment of these constructions. So could that be an explanation of why when you introduce deletions in the middle, you stabilize the structure because the dipolar moment is lower? And second, this usually is counter-affected by the distribution of charged amino acids in the extremities of the helices. This could be some of the mutations that could stabilize the thing?
Yeah, it's confusing, but maybe, yeah. I wish I gave this talk earlier so I could spend more time talking to you.
So Bob, in P22, the key aspect of the scaffold is dimerization. Do you know anything about the oligomeric state when you isolate the scaffold? Could you express the scaffold? And
We've expressed the scaffold, and it went to George Thomas's lab, and it's been...
But I mean, like, oligomeric status, did you do any centrifugation on it?
We tried to, we collaborated with Angela Gronenberg, tried to do an NMR structure, and so we made some labeled delta domain, purified it, and she said, it's a soluble aggregate.
Okay,
Goodbye.
so it makes so it makes larger oligomers.
It makes, yes, mixed bag.
Okay.
Somebody suggested I should have looked at it in the EM, but I never have. Yeah. Not in control.
It's great to end a meeting on a symmetry controversy, stoichiometry controversy. But it seems like your deletion mutant suggests that you're changing registries, right? Like if you delete 7, it's like it can reorient itself and make the proper oligomer because you've rotated the whole helix around one turn, basically, and glued it back together.
Yeah.
But the intermediate ones with the T equals 4 are of a different registry, almost as if you're shifting the stoichiometry of the coil from one version to another, like from a trimer to a dimer, and so therefore it can recover that, it's switching registry and oligomerization state. Am I crazy?
No, you're not crazy. Just tell me how to do the experiment.
Great work, Bob.
Yeah.
All right, so let's thank the speakers in this last session for great talks. And before I hand it over one last time, I think we should give our